just a short note regarding the previous very interesting discussion, which touched regarding two issues. Standardization, how can I be sure that I will get the same treatment in different centers? And second, the cost. Affordability, how can I afford these costs? An answer will probably come from automation. Several companies are investing in automation, resulting in the production of systems which are standard, resulting in increased standardization, ensuring that everybody can receive the same kind of product. The process is the product, it was said before. And also with the production of bioreactors, disposable bioreactors, which will reduce the costs and decrease the production time. Now, our next speaker in the flash talk session is Dr. Andrea Belluati from Darmstadt, Germany. The title of his talk is A New Skin, Self-Synthesizing Artificial Cells via Enzymatic Polymerization.
Right, so thank you for the invitation here, the introduction. I promise I won't keep you long away from your coffee. Just a very quick reminder of where Darmstadt is for those who don't know it. We are here in the center of Germany. This is where we are lost in the forest, and this is our tiny group that has been working on this together with me. Luckily today we already introduced the concept of bottom-up synthetic biology and why we want to work our way up from non-living components to components that imitate life to the point of which we hope we might at some point cross the threshold between abiotic matter to living matter. So this transition from abiotic to biological chemistry has been posited to be based on the existence of compartments, of containers that allow the division between a non-living and a living system, anyway, a system that can evolve to that point where you get to an actual biochemistry. This is called the GARD system, and on this topic we have developed the concept of artificial cells that today, luckily, was already introduced by previous speakers. So artificial cells are objects that look and behave at least in one way or another to living cells. Now, usually when we talk about living cells, we want to use compounds, chemical compounds that are biologically derived or are found in nature. And if you're talking about the container, what drives the compartmentalization of life, we tendentially use the same components of our cells. So, for example, the lipids that our cell membranes are made of. And we can also make a simplified system, which is called the liposome, has been already used in many different applications already in the pharma industry, for instance. However, we could think of using synthetic molecules that behave much like the lipids in the sense they self-assemble into these 3D structures, and they are called polymersons because they come from synthetic polymers. Polymersons, compared to liposomes, have the same capability of encapsulating hydrophilic and hydrophobic molecules. They can be engineered to be biocompatible, but on the other hand, they tend to offer higher stability and better versatility in chemical terms. Now, the idea of making cell-like systems out of polymers is not new. It has been already shown that you can indeed have polymersomes looking like artificial cells made out of lipids if you encapsulate the same machinery that leads to the production of proteins, for example, in synthetic cells. Now, this leads to the question, okay, we can do it, we can be in a lab, we can make polymers and with organic chemistry and make this kind of polymer like artificial cells. However, can we make this material, can we produce this material in a more biosimilar way? Here we use this concept of polymerization-induced self-assembly that, very much like the phospholipids of the membrane are synthesized, starts from two water-soluble molecules that, as they are synthesized, become amphiphilic and then lead to the construction of these 3D structures, from nanoparticles to vesicles, that is, 3D structures with no aqueous lumen inside. In this case, for this pizza, you can even see it, if you do it via photoorganic chemistry, you can see these vesicles grow and then expand until they burst and grow again. However, again, this is based on classical organic chemistry. What if we used enzymes, that is, the natural catalysts, to produce these polymers? And we can indeed use some proteins, namely metalloproteins like myoglobin, hemoglobin, peroxidase in general, to lead to these polymers that self-assemble into structures. Hence, with my collaborators in Switzerland, we developed this enzyme biocatalytic polymerization-induced self-assembly using myoglobin, a typical protein found in muscles, for example, that goes from a water-soluble polymer and monomers that are water-soluble to this amphiphilic molecule that self-assembles. This is how it looks like chemically, won't go into detail, but long story short, we can find structures thanks to these non-biological polymerizations that sort of look like cell membranes. And, for example, we can even get enzymatic reactions inside. This is a classical one where you have two polymers. One is the catalyst, which is self-encapsulated, hence we call it self-synthesizing, that can, for instance, produce a fluorescent compound such as zorophin. You can see that it gets produced inside these vesicles. But beyond that, if from just some simple enzymes we put the full machinery of a cell, for example, an E. coli bacterium that goes with all the cell lysate, it's basically a non-defined soup that also has ribosomes and all the other enzymes necessary to go from a plasmid, so DNA, to a protein. If we feed from the outside the building blocks, nucleotides and amino acids, we can, for example, produce a fluorescent protein inside these cell-like structures. And what is also interesting is that this polymer tends to stabilize these macromolecules that would otherwise tend to stop working after some time at higher temperatures, and by higher I mean fridge temperatures, whereas here we can still see some activity after 1 year in the fridge. So, to wrap up, we do have, thanks to this biopolymerization-induced self-assembly, structures that look like cells that can be produced using a non-biological but actually biochemical polymerization. We can encapsulate macromolecules, enzymes, any kind of protein, DNA, and whatnot. And by going to a complex cell lysate, so an extract of cells, we can have these cells somehow be revived, if you allow me this expression, inside a non-biological membrane that can then produce, for example, proteins. But this leads us to some new questions, some questions that might be even ethical besides definitely being philosophical in origin. So, materials. What kind of materials that are not found in biology can behave somehow like life we see today on Earth? And to what extent can we go to that point? Do we actually need some specific biological components, compounds to do that, or can we actually do it with novel molecules never seen before on Earth? And what happens in this case, should we get to that point, once we get a new matter that behaves like life? What are we doing? What are we making? Are we making new living systems or just systems that somehow look like they're alive, but much like a robot could look like a person, but will never be one. So I will finish here my presentation, my flash. Just want to thank you, the audience, my collaborators, and my funding agency for the support for this work. And thank you for your attention.
Thank you, Dr. Belluati, for this outlook into the future. Time for a short question.
I have one. How do you feel this mechanism is similar to what we discussed about digital twins? Because actually this is exactly the same process, but with some more nuances and possibly perspectives. And do you feel like these two things might be integrated together, digital twins and artificial cells?
Yeah, I would say that yes, definitely this is something that could be the matter counterpart to these digital twins. Our saving grace that has saved us from ethical considerations so far is that we are very far away on a mechanistic level from behaving like life. So we've just been pushing it forward to the future. But yes, at some point you could think of having non-biochemical twin to actual life, and that will be something to be discussed on in the future. Thank you.
Thank you. Just very shortly, do you foresee a future for artificial cell therapy?
Yes, definitely. Not necessarily this specific material I have been using. There are many different approaches to that, but in a way you are simplifying the very complex systems of life without necessarily shaving off too much of the complexity you need to get more emergent properties. So I do hope that this kind of approach might lead to real-world applications in the future, by my approach or others that have been presented, will be presented in these days here, for example.
Thank you. Thank you very much. Our next speaker is Dr. Samuel Santos Gomez from Augsburg, Germany. The title of his presentation, Role of Surface Phase Transitions in Biology.
So, very good afternoon, everybody. Again, sorry for delaying for the coffee, but I hope I still have your attention. My name is Samuel. I'm a PhD student, not yet a doctor, again, but hopefully soon. I'm a biophysicist, and I'm coming from Augsburg. You've already seen a talk from one of my colleagues. I've titled my talk The Role of Surface Phase Separation in Biology, and I'd be happy to talk about this more in detail during the conference, but you'll see that the talk that I prepared today is much more general and wants to address the bigger picture and the larger scope of the ethics of engineering life. So my colleague Gaetano already mentioned this point, that this idea of synthesizing life or engineering life comes with the other side of the coin of ethical issues and responsibilities. And coming from a theoretical physics background, I think it puts us in a very interesting position to discuss this topic. Why do I say this? Because among all the fields that are needed to collaborate with one another to answer such a scientific endeavor, I think theoretical and computational biophysics, or modeling in general, acts as a very good linker and communicator between these different fields. Theoretical physics, or theory in general, has this overarching goal to describe the laws that govern the functions of these different fields in mathematics. But I think, given the topic of this conference, I think we can also play and have a responsibility to play a role in ethical discussions in the field. Why is this so? I mean, I think one powerful aspect of theory is that it has this ability to extract general principles and generate testable predictions. I think this is very important because having testable predictions or anticipating them helps us to prepare ethical discussions and regulations based on the consequences of these predictions. And I think we already see it in the world today with artificial intelligence, is that oftentimes regulation doesn't go at the same speed as innovation. And this is an important aspect where theory can come in to help. So these words, actually, maybe it's divine intervention, but all the flash talks seem to complement one another quite well. But you've seen these two approaches that are used. They have been repeated throughout the flash talks, and they are general approaches that are used in science. And they're very useful for answering a question like engineering life. The bottom-up approach, which essentially starts from very simple building blocks and progressively increasing the complexity of the system to eventually lead to something that is relatively complex, that has some emergent property. And this is a very useful method. But you can also do the opposite and look and use the top-down approach, where you start from trying to look at a very complex biological system and then reduce it component by component to the most minimalist model possible that still performs the function that's necessary. So my colleague Gaetano already mentioned one example of a model that is based on the bottom-up approach, where we start with simple building blocks and progressively increase the complexity to create a model for genome replication and evolution. And today I just want to walk through an example of the top-down approach in the context of intra-extracellular communication. So the question is, why are we looking at cells? Well, if we want to look at the top-down approach, we have to start at the most basic unit of life as we know it. And this is the cell. And it's a very complicated component with a lot of machinery. And the real question is, and this is not an easy one, is among this living system, which I think there's a consensus that a cell is living, what is the minimal subset that we need to extract from this to create something that is still considered a living system? In fact, there was a scientific discussion in a conference last year that a lot of scientists here were involved in, and there's a paper that is I recommend to read. It was about a roadmap towards the synthesis of life, and we had a consensus on this definition. So we defined synthetic life as a self-sustaining chemical system from non-living matter capable of open-ended Darwinian evolution. Now, I'm not saying this is definitive, and I don't think any of us are or will. It's still an open discussion, but is this enough to really pave the way to engineering life? So there are hypotheses that there might be other ingredients that are involved, and I just want to walk through one example of it. But essentially, some of the key ingredients might be a self-replicating system that is able to compartmentalize, have some kind of metabolism. And if you put this in an environment that is out of equilibrium, that is capable of Darwinian evolution, possibly we can get synthetic life. And I just want to stress now the role of compartmentalization. So compartmentalization occurs naturally in the cell, and it's one of its basic functions is to have a physical barrier that distinguishes the physical self from the environment. It also has the usefulness of preventing components from just spreading into the environment. It can also act as microreactors or a mechanism for protecting components. There are two classes of compartments that you find usually, and they've both been talked about today to some extent. There are some membrane-bound components that we just saw in the previous talk, compartments that are encapsulated by a membrane. But then you also have membrane-less compartments, essentially compartments that are made of possibly similar liquid-like materials as the cytoplasm of a cell or whatever environment, but have distinct physical properties that are different to its environment. And one physical process that can lead to it is phase separation. This is much like mixing olive oil in water and seeing that it doesn't mix. And this occurs also in the cell in many different processes. I mean, here is just a few examples highlighted in purple of membrane-less organelles, but also membrane organelles within the cell. And it's very important. The human cell today has both of these as necessary mechanisms for compartmentalization. So let's just take a quick focus on the membrane as a compartment. So it's a very complicated thing. There are a lot of proteins and different components that are diffusing around, binding. It's a very complex system. And what I want to just talk about is this idea of how do cells communicate through a physical membrane? And how can we look at this or simplify this complex system? One of the functions that a cell needs to communicate to the other when they come in contact with one another is the formation of a tight junction. Very briefly, it's a protein complex whose one principal function is to create a tight seal between epithelial cells. This is why if I put a drop of water on your skin, it doesn't just leak in between the cells. It has to either pass through diffusion or osmosis or through some active process through a membrane protein. And this is a very complicated process. When two cells come in contact with one another, there's a lot of binding, diffusing, in fact also phase separation on this 2D surface of a membrane that lead to the formation of such a protein complex junction. But what we can do with the top-down approach from a theory point of view is to try and look at this complex system and identify the key ingredients that are needed and the key functionalities needed, and reduce the system to just a few components that have certain functions like binding and phase separation, or the ability to phase separate. And this is one example of how theory from a top-down approach could be used to help. So I end my talk here, slightly abruptly, but I want to leave you with a few conclusions. The first is that I think as theoretical biophysicists and theorists in general, we should help anticipate ethical discussions before they actually become a reality. I think it's an important role that we can play in this discussion. Also, using both the top-down and bottom-up approaches helps us to better understand the different design elements that are needed for engineering life, and both can benefit from one another. Also, compartmentalization could be a key aspect in engineering a synthetic cell or engineering life. And lastly, the importance of collaboration and communication as a community, which I think this conference is a great example of. So with that, I'd just like to thank you for your attention, to the organizers and to the funding. Thank you.
Thank you, Dr. Gomez, for adding another piece to today's puzzle on the what is life question. Time for a short question. Yes.
Thank you. Can I ask the definition that you had of life was very interesting.
Yeah.
But apart from self-replication, which is definable easily, how do you define the other bit? What constitutes the other stuff? And this is the word autopoiesis, for example, which I'm sure you know. Is that involved in the other stuff?
It's a great question, and I don't think I'm the most qualified person in the room to answer it. But I think, so self-replicating, as you said, I think it's one aspect that could be useful, but if you see here, actually replication is not really used in this definition. And an example of this, let's say, discussion that we had was this consensus on the term self-sustaining chemical system. One big example that was brought about a lot are viruses. So viruses don't have their machinery themselves to replicate, but they need the machinery of the host, but they are living entities. The other stuff, so open-ended Darwinian evolution, I think this term comes with a lot of weight, because to define what open-endedness means, even in our field, is, I would say, not so clear. And Darwinian evolution also, I think, comes with its own caveats when you look at it from a molecular level, because sometimes it's a bit difficult. For example, in Eric's talk, we talked about information carriers also being structural components. And this is one example of a genotype acting as a phenotypical role or something like this. So definitely, I don't know if I have the answer, but these are just some of my reflections on why this could be a good discussion to have.
Thank you, Dr. Gomez. Now our last speaker, last but not least, of course, is Dr. Florian Schmidt from Zurich, with a puzzling title, Record Sec.
That works great. We've had a lot of talk on how CRISPR could be used to edit the germline or edit diseases in human patients. I'm going to try to convince you that CRISPR can also have entirely different purposes. And what I'm going to go for is these little bacteria here. They usually reside within your gut. And within these bacteria, we have found a particular CRISPR system that we have turned into what we call a molecular video camera. And our vision is that eventually we can take this system back into your gut and monitor what you've had for dinner last night, because probably you haven't been having what your doctor recommended you to have. So let's get into it. Kind of the advent of this project, which I started doing my PhD, was RNA-seq. This is a high throughput technology that really enables us to investigate the transcriptome and thereby the behavior of cells. And it gives us an incredibly rich picture, but it has one major limitation. To investigate the transcriptome of a cell, you have to destroy it. And this situation, it's very much fine if your intention is to make carbonara, but it's not fine if you're thinking about your experiment where you're looking at a cell and that cell might continuously adapt and change to what's happening around it. And as a consequence of that, also the transcriptional state of the cell adapts. Now, if you're using RNA-seq, which we like to compare to that first generation snapshot, you get a very rich picture at the time point where you're sampling. But you're missing on anything that happened before, and you're also missing on anything that happened thereafter. So what we decided to set out to do is to create the molecular video camera, something that sits within the cell and keeps track of these transcriptional events, records them back from RNA, which is this transient form of information, back into DNA, somehow storing them inside the genome of the cell, so that in the end, this information is inherited from one generation of cells to the next, and we can easily retrieve it in the end and tell you about everything that's happened and not just about that one singular event. So is there any way that bacteria can already do this? And turns out this is where we get back to CRISPR, this adaptive microbial immune system that creates this vaccination card within the genome of the bacterial cells, as my professor Andy Platt has elaborated on this morning. So what happens here is you have this bacteriophage. This is a virus that infects bacteria and that injects its own genetic material into a bacterial cell to coax it into doing what the bacteriophage wants. And the bacteria have figured this way via CRISPR to store short pieces of the genetic material of that bacteriophage within their own genome. So this gives them information about their invaders and eventually allows them to defend themselves. And we thought, well, that's cool. That's a molecular recorder. So can we use this? And turns out there is a molecular machinery behind this. So you got these two CRISPR-associated proteins that find each other, and then they bind a piece of nucleic acid, and they are ready to integrate it into the CRISPR array. Now, most CRISPR systems, they record their spacers from DNA. So that doesn't quite do what we wanted to do. But in 2016, scientists at Stanford, they found a particular flavor of this Cas1 protein. So that's one of the proteins involved in this protein complex that's naturally fused to reverse transcriptase. So that can take this transient piece of information, the RNA, and record it back into DNA. And so what we postulated based on this discovery is that you could take this RT-Cas1, Cas2 complex, you could make cells express it, and then these cells, they would continuously sample the RNAs that are within them, and they would integrate that information into the DNA where we can conveniently read it out in the end. And thereby we could keep track of these changes that happen to the cell. So nice for the theory. What do? We take these cells, they express their recording machinery, we expose them to all sorts of different environments, and in the end of the experiment we can extract these spacers. These are these short snippets of RNA that contain information, and we can map them back to the genes of their origin. So we figure out what the bacteria have been up to, how did they adapt to the conditions that they have been exposed to, and we can generate these count matrices where you get the number of genes that have been expressed under each particular condition. And then we can do this to perform our analysis, to perform correlation, clustering, differential component analysis, and hopefully if we use that in the human gut, tell people what you have been eating. But first things first, did we actually create a molecular video camera, or are we just doing something that RNA-seq could have done long times ago? Or in other words, can record-seq, can this technology capture transient signals and preserve that information over time? And so the experiment that we set out to test this is pretty simple. You take 3 test tubes with bacteria and one just serves as a control. These bacteria, they are not put under any stress, they can live happily. And the second group serves as another control. These bacteria are stressed. We add a herbicide to them and they will be provoked to respond to that. And then we have another group that also sees this herbicide, but only for a short period of time. And then we use both RNA-seq, so the traditional high-resolution method that captures a snapshot, and record-seq, so what's hopefully our molecular video camera, and see what we can observe from these three different conditions. And as you would expect, both RNA-seq and record-seq, they can distinguish these two extremes. So they can distinguish cells that have never seen the stressor from the cells that have permanently seen this stressor. But where it gets interesting is where do these cells end up? The cells that have seen the stressor, but only for a short period of time. And it turns out in RNA-seq, they cluster completely with the unexposed groups, meaning that the transcriptional signature of the cells adapting to that stressor, it's gone. The cells readapted to the medium that doesn't contain the stressor and they're happily living. You can't distinguish them. With record-seq, it's the exact opposite. These cells, they cluster with a permanently exposed one. So our molecular video camera, it was able to record this information into the DNA. And in the end of the experiment, when RNA-seq was long unable to detect this, we're still able to recapitulate this information. So that's all bacteria in a test tube. How do we take this to the real life? And that's essentially where we decided, well, this could be a great tool to interrogate the gut microbiome. And the idea here is that the microbes that reside in our gut, they constantly adapt to what we do to them, what we eat, whether we're stressed, whether we get enough sleep, and even the interactions between our gut microbiome and the brain. And if you were to observe this, you had two different methods. One would be breaking the egg. So in a human, that means you need to perform a surgery and get them out there, which is not really what you want to do unless it's absolutely necessary. And the second one is you have to rely on fecal measurements. Now, fecal measurements have the problem that they are taken at the very end, and the microbes have adapted a lot already. And what you wanted to recapitulate from the early parts of your intestine has been lost, very much like the stressor in the previous experiment. So the rationale here is, can we feed our bacteria first to mice and hopefully eventually to humans and see what happens within the intestine and get an idea about this black box? So that's just what we set out to do. We take these bacterial sentinel cells that record what they adapt to in their environment, and we feed them to mice. And then these cells, they'll traverse the gastrointestinal tract, and they'll observe all sorts of different environments. They'll encounter whether you had an inflammation, they'll also see what you've been eating, and they'll adapt to that. And our molecular video camera, it sits within that bacterial cell, and it keeps track of these things. So hopefully in the end of the experiment, we can sample the feces, we can perform our record-seq method, and we can obtain the information that these bacteria have been recording. And I should highlight that this was part of a larger collaborative effort. So not only my PI, Randy Platt, and another computational PhD student in our lab was involved, but we also worked together with experts in the gut microbiome, namely Professor Andrew McPherson. And what we found essentially when we start to feed these bacteria to the mice is that as we progress in time, we accumulate these spacers, meaning our molecular tape recorder, it accumulates information that we can read out in the end. So that was really promising, but it's just a general example of we're recording things. So let's get deep. Let's get into the host's diet. Can we detect whether you've been having all'amatriciana or carbonara yesterday night? And we're not exactly feeding that to mice, but we're feeding 3 different diets to mice. And in this case, we fed the mice for about 1 week, and they also contained our recording bacteria. And we again use RNA-seq and record-seq to analyze the situation. And as you would expect, RNA-seq, it's perfectly able to distinguish these 3 conditions. Record-seq, same picture, we can distinguish the 3 conditions. So far, so good. Where things get a little more complicated is when we ask, can they also preserve information over time? And what we did to test that is we continue the experiment, but now we put all the mice on the same diet. This is about the same thing that's going to happen to a lot of you after Christmas when you have your New Year's resolution, and this is where you start to eat healthy again. And of course, that's what you're going to tell your doctor you've been doing all year long. And if your doctor only had RNA-seq to look at your gut microbiome, things would look like this. So again, we're looking at this dimensionality reduction. Samples that are closer to one another, they'll cluster with one another. And samples that are more distal to one another, such as these small dots in the different color, they are further apart. And the additional element that we bring in here is that the size of the dot corresponds to time. So what you see here is that in the samples that are taken very close to that moment before you change the diet, they are still distinguishable. But as soon as you switch the diet, these dots, they start to converge. And a few days after you went to the healthy diet, you're unable to distinguish the situation if you only had RNA-seq. While with record-seq, things are a lot more clear. You can see that even 2 days after we changed the diet, we can still distinguish the mice that have been eating healthy all year long from the mice that have been on these 2 different diets. And with this, I would already come to the end. I would like to highlight that the gut microbiome is insanely complex, and that with record-seq we can hopefully in the future shed a little bit more light on what's happening in your upper intestine and what diet you've been eating. And I would of course like to thank all the fantastic scientists that have been involved in this work and that also climbed mountaintops together, and I'm happy to take any questions.
Thank you very much, Dr. Smith, for this clear presentation. Question?
Sure. Thank you. Quick question. That's fascinating to do that. How much perturbation do you think your record-seq has on the metabolism of the E. coli itself that throws off your...
Great question. I would wish we were actually perturbing the E. coli cells. The system has evolved to be very inefficient, so it's not a single cell method. There's spacers acquired in every about 1,000 cells. So it's a population-based measurement. In the future, you're of course completely right. If the system becomes more efficient and it starts to, chop up RNAs and integrate them into the CRISPR array, that has profound consequences because you're influencing what the bacteria are doing, and probably even more importantly, you're massively expanding their genome, and that eventually becomes a cost. We're far from there.
I think you just answered my question, but do you have any temporal resolution of this? Can you tell a series of events, or you can't because it's only recording 1 in every 1,000? So, yeah.
Very close to the previous one. So this particular flavor, this RNA recording CRISPR system, it's at this point too inefficient to get a lot of temporal resolution, but we're not far off. There are DNA acquiring CRISPR systems where people have gotten a temporal resolution.
And can it only pick up internal RNA sequences, or are you picking up exosomes or anything else from the host organism?
Exosomes in bacteria, I'm not sure if that's a thing, but yeah, but I'm not sure how much that they would take up. In principle, we have looked for sequences that are not the E. coli genome or the plasmid we use to overexpress this, and we haven't found much. So for now, I would say no. But we can, for instance, record the invasion of a phage into the bacterium.
Thank you again. Coffee time.
Okay, I suggest